Hello, my name is Kevin Warner. I'm an associate professor of entomology in the Department of Plant Sciences. I'm also the extension specialist for cropland entomology. What that means is that I advise farmers in Montana on managing insects that are feeding on and damaging their crops. For research, I work on applied research topics in terms of managing these insects that are damaging crops, but also I have a basic research component that involves insect molecular biology. So for several years now, I've been providing this lecture for David's class on insect biotechnology. And the lecture has evolved over several years. Insects are very diverse, and originally I would take a broad approach to this class. But a lot of interesting biotechnology is taking place with mosquitoes right now. So the lecture has come to focus on mosquitoes, and for good reason, as you'll see. I'm going to start off the lecture with a broad introduction to insect diversity for those of you who aren't familiar with insects, and then move into mosquitoes as a vector of human diseases. Insects are very diverse and they've evolved many different feeding habits. They live in many different environments. So blood feeding is one of the mechanisms that insects have evolved. Most of you have run across mosquitoes, and as blood feeders, they vector many important human diseases, including dengue fever, Ross River fever, malaria, which is probably the most important, Zika. So for that reason, blood-feeding insects are very important to us as medical pests. Insects are very old. They evolved over 420 million years ago. They're also very numerous in terms of the number of species. Beetles alone are estimated to have over 1 million different species globally. So with that large number of species, as well as over 400 million years to evolve, insects are very diverse. They've evolved many different body forms, many different feeding types, and many different habitats. Looking at mosquitoes as medical pests that vector important human diseases, mosquitoes are also diverse. There's 40 different genera globally with 3,457 species. So mosquitoes themselves as blood-feeding insects are also very diverse. And if you look at the diversity, of course, there's greater diversity in the warmer climates. This chart simply lists some of the important human diseases that mosquitoes vector and where they're located across the globe. Of course, malaria is very well known, and it's one of the most important mosquito vectored diseases. And because it's one of the more important mosquito vectored diseases, there's been a lot of research that focuses on malaria, including biotechnology. So we'll see a little more information about that in this lecture. One point to consider, we've seen that mosquitoes, there are many different species of mosquitoes, several different genera. Multiple species of mosquitoes vector human diseases. So many different mosquito species vector these human diseases. And also a single human disease can be vectored by multiple mosquito species. So again, if we're looking at malaria at the top of the chart, malaria is caused by the pathogen Plasmodium, but there's several species of the pathogen Plasmodium. As well, this Plasmodium pathogen can be vectored by species of Anopheles, as well as Culex species. So it's not a simple matter of a single mosquito species and a single pathogen, so the problem can be a little more complex. YouTube has great videos, great material. And in my past lectures, I've used these videos. I pause and I show these videos. So at this point, I'm going to pause and play some videos that illustrate the life cycle of mosquitoes as well as the mechanism by which they feed. This first video is an animated video of the mosquito life cycle. So of course mosquitoes lay their eggs in water. The mosquito larvae are aquatic. The larvae are filter feeders on the surface of the water. And then they'll go through a pupal stage where they then again emerge as the adult mosquitoes. So that was an animated video that I thought was kind of cool. This one here will be narrated.
Mosquitoes go through 4 stages of development in their lives. They undergo complete metamorphosis. Culex mosquitoes lay
Steve Daines always follows the money.
Of fresh water, or in areas that will be temporarily wet, like floodplains. The eggs float alone, or in groups that look like rafts. Eggs laid during winter can survive to hatch in the spring. Mosquito eggs hatch into larvae. The larvae live at the surface of the water and breathe through a tube. As the larvae grow, they shed their skin a few times. If mosquito larvae need to hide from predators, they can swim and dive below the water's surface. Mosquito larvae live a few days to a few weeks before they turn into pupae. They stop feeding just before they turn into pupae. The pupae live at the water's surface and breathe through 2 small tubes. Mosquito pupae do not eat, but they are active. They tumble below the surface of the water when disturbed. They remain in the pupal stage anywhere from 1 to a few days, depending on the water temperature. While inside the pupa, the larva transforms into an adult. The adult breaks out of the pupal case and crawls onto the surface of the water. It rests while its exoskeleton hardens. Later, it crawls to a safe place out of the water and spreads its wings out to dry to prepare for life on land. The mosquito is now a flying land insect. Adult mosquitoes mate to produce eggs for the next generation. Adult female mosquitoes can live from many days to weeks, while adult male mosquitoes usually live just a few days after they mate.
All right, one more video that shows specifically the mechanism, the feeding mechanism of mosquitoes.
This is the deadliest animal in the world. Mosquitoes kill hundreds of thousands of people each year. The most vulnerable people, children, pregnant women. No other bite kills more humans or makes more of us sick. So what makes a mosquito's bite so effective? For starters, they're motivated. Only females bite us. They need blood to make eggs. And a pool of water for their babies to hatch in. Even a piece of trash can hold enough. At first glance, it looks simple, this mosquito digging her proboscis into us. But the tools she's using here are sophisticated. First, a protective sheath retracts. See it bending back? If you look at a mosquito's head under a microscope, you can see what that sheath protects. And inside there are 6 needles. 2 of them have tiny teeth. She uses those to saw through the skin. They're so sharp, you can barely feel her pushing. These other 2 needles hold the tissues apart while she works. From under the skin, you can see her probing, looking for a blood vessel. Receptors on the tip of one of her other needles pick up on chemicals that our blood vessels exude naturally and guide her to it. Then she uses this same needle like a straw. As her gut fills up, she separates water from the blood and squeezes it out. See that drop? That frees up space to stuff herself with more nutritious red blood cells. With another needle, she spits chemicals into us. They get our blood flowing more easily and give us itchy welts afterwards. And sometimes, before she pries herself away, she leaves a parting gift in her saliva. A virus or a parasite that can sicken or kill us. There's nothing in it for her. The viruses and parasites are just hitching a ride. But this is what makes mortal enemies out of us and mosquitoes. They take our blood. Sometimes we take theirs. But often not soon enough.
Okay, so some interesting videos on the mosquito life cycle and how they feed. And you can see how, of course, some people thinking a good insect is a dead insect. But for those of you who aren't disgusted with insects, you can see they've really evolved some pretty amazing lifestyles and morphological structures to get done what they need to do. So we're going to move on now. This was an introduction to mosquito biology, mosquito life history, and we'll go on and talk a little bit more about malaria and efforts with biotechnology to reduce the amount of malaria that mosquitoes can transmit. If you're looking at preventing a disease, you look at this disease triangle. There's an interaction between the environment, the vector disease, the pathogen, and the host. To prevent disease, you can target the vector, the pathogen, or the host. And of course, with the biotechnology approaches are looking at creating genetically modified mosquitoes. And the approaches are looking to genetically modify these mosquitoes to either reduce their populations, so there aren't as many mosquitoes to vector the disease, or these approaches are looking to modify the mosquito to either kill the vector within the mosquito or to engineer mosquitoes that are less able to transmit the disease vector. So those are some of the strategies that biotechnology is taking to try and reduce human diseases vectored by mosquitoes. I'm going to stop again, and we're going to focus on malaria, which again is the disease vectors Plasmodium. And I'll show you a brief video about the life cycle of malaria so that you can understand how some of the biotechnological approaches with mosquitoes are focusing on different different stages of the Plasmodium itself.
Human skin to obtain a blood meal. It injects saliva mixed with an anticoagulant. If the mosquito is infected with Plasmodium, it will also inject elongated sporozoites, motile, spindle-shaped asexual cells, into the bloodstream of its victim. The sporozoites travel to the liver, where they enter liver cells and rapidly divide asexually. This asexual division, which is called schizogony, generates the next life cycle form, called merozoites. The released merozoites invade other liver cells and enter the host's bloodstream, where they invade erythrocytes. Once inside the erythrocyte, the merozoite begins to enlarge as a uninucleate cell termed a ring trophozoite. The trophozoite's nucleus then divides asexually to produce a schizont which contains several nuclei. The schizont then divides and produces mononucleated merozoites. The erythrocyte ruptures and releases toxins throughout the body of the host, bringing about the well-known cycle of fever and chills that is characteristic of malaria. Plasmodium enters a sexual phase when some merozoites in the erythrocytes develop into gametocytes, cells capable of producing both male and female gametes. Erythrocytes containing gametocytes do not rupture. Gametocytes are incapable of producing gametes within their human hosts, and do so only when they are extracted from an infected human host by a mosquito. Within the gut of the mosquito, the gametocytes form male and female gametes. The resultant diploid zygotes develop within the mosquito's intestinal walls and ultimately differentiate into oocysts. Within the oocysts, repeated mitotic divisions take place, producing large numbers of sporozoites. These sporozoites migrate to the salivary glands of the mosquito, and from there are injected by the mosquito into the bloodstream of a human, thus starting the life cycle of the parasite again.
All right, from the video, you could see there were a couple of stages of the Plasmodium life cycle that were critical in terms of how it interacted with the mosquito. So there was a stage where the Plasmodium had to cross the insect gut. And then there's the stage where it has to get back into the salivary glands, because the salivary glands will be the mechanism for transmission during a blood feeding meal. So, what are some of the biotechnology approaches that have been taken to try and prevent these steps from occurring? As was mentioned before, just to review again, Plasmodium, there are 5 different species, and these different species of the Plasmodium disease are carried by the genus Anopheles, which has 50 different species of mosquitoes in that genus. Again, a little more background information. Mosquitoes are estimated to transmit disease to more than 700 million people annually, killing at least 2 million. And one of the reasons why we're going to focus a little bit later on malaria is because up to 1 million people are killed by malaria each year. So it's one of the more significant diseases vectored by mosquitoes. So, insect biotechnology. We're talking about genetically modified mosquitoes, GMMs. And when we looked at that disease triangle, you can target the pathogen to reduce, target the vector. You can target the vector to reduce the population size or prevent them from transmitting the disease. And so that's the strategy that these genetically modified mosquitoes, or the research towards this, so these don't exist, well, there have been some trials that you'll see at the end of the talk, but right now what we're talking about is research that's occurred over the last 10 or 20 years to create these genetically modified mosquitoes. One approach in the laboratory has been to block receptor proteins in the gut. So again, remembering there's that stage of the Plasmodium that has to cross the insect gut. So there are receptor proteins that are recognized by the Plasmodium that allow it to transfer across the gut. So if you can block these receptor proteins, create a genetically modified mosquito where these receptor proteins are blocked, you prevent the Plasmodium from completing its life cycle because it can't get across the gut. Another strategy has been to genetically engineer mosquitoes that produce an antibody. It's not an antibody that it would normally produce, remembering antibodies are proteins. So if you can introduce the gene for that antibody into a transgenic mosquito, and this antibody targets the parasites and kills the parasites within the mosquito. So these are our 2 strategies that have been investigated in the lab, but these 2 strategies have not gone forward. And one of the things you're going to see during this lecture is just how quickly the technology is changing, how quickly biotechnology is changing. There were probably, I'm guessing, but probably a decade of research went into these 2 approaches, these 2 strategies. And then new technology emerges that's more promising. And so we're just seeing a very rapid change in the technology and biotechnology. One technique that was developed in agriculture is termed the sterile insect technique. So essentially, the idea of the sterile insect technique is that you raise in the laboratory or an insect rearing facility millions of male insects that are sterile. And then when they mate, you release them. And when they mate with the wild females, no offspring are produced because the males are sterile. So overwhelming numbers of sterile male insects are released, and they compete with the wild males. And if you do this repeatedly, then you can suppress the population. And there have been 1 or 2 examples in agriculture where they were able to eradicate that insect from a particular region. Originally, this technique used radiation to sterilize the male insects after they had been reared in a facility. That can cause problems with the fitness of the males. They may not be, the sterile males may not be as competitive with the wild type males, and it's also very expensive. So again, just a schematic to illustrate how this sterile insect technique would work. You're rearing millions of insects. You're sterilizing them, usually with radiation. You're releasing them over a wide area. And on the right-hand side of this slide, you'll see the sterile males are represented by a red square box. So the native females that mate with the sterile males, they produce eggs, but the eggs aren't viable. So again, in that schematic on the right-hand side, you can see a sterile male illustrated by the red box mating with a wild-type female, and you see the red offspring have an X through them because, well, there are no red offspring. The eggs were not viable. No larvae hatch from those eggs. You still have some wild-type males and females that are mating in this population, though. But the idea is that R1 release 1, R2 release 2, R3 release 3. So if you continue to release millions of these sterile male insects over and over again, they're going to become more and more prevalent in the population, and the probability of the wild-type females finding a wild-type male to produce viable offspring goes down. And eventually, using this technique, you can either suppress the population so there's low numbers of the pest population, or if there's a restricted geographic area, you can actually eliminate them from that geographic area. Now, where does biotechnology come into this? Well, as I mentioned, this technique of the sterile insect technique was originally developed in agriculture many years ago. Biotechnology was very early or didn't really exist at that point. Now, with the recent advances, we have Oxitec, which is a private company, a spin-off of Oxford University Research. And they've used biotechnology to develop a transgenic strategy for creating sterile insects. And they've applied this to Aedes aegypti males in an attempt to control dengue fever that's vectored by this mosquito. So they're taking this principle that was developed in agriculture, where it wasn't very effective because you had to sterilize the insects with radiation. They've now applied biotechnology to create a genetically modified mosquito where the males are sterile. Now, how have they done this and what is the technology? They're calling it RIDDLE, release of insects carrying a dominant lethal gene. So they've created transgenic mosquitoes where the males carry a dominant lethal gene. This dominant lethal gene, it's lethal to the larvae. So the transgenic males compete with wild males. They mate with the wild females. But those offspring, those larvae, carry this dominant lethal gene and they die as larvae. Now you might ask, how do they raise these insects if the larvae carry a lethal gene? Well, there's a mechanism, a rescue mechanism, so that while they're rearing these transgenic mosquitoes, the larvae are rescued, they're not dying. So Oxitec has been conducting trials to determine whether or not their sterile mosquitoes can actually reduce the populations of the wild mosquitoes and reduce dengue fever, the disease vectored by these mosquitoes. And they've now conducted several trials worldwide. Now, as you might imagine, this is controversial. It was kind of interesting because last year I surveyed the class, and it seemed within the class the idea of releasing a genetically modified animal, a genetically modified insect into the environment was not so controversial to the class when I surveyed them last year. But if you look in the general public, there is some opposition to this. These trials so far have been conducted in the Cayman Islands in 2009, where Oxitec released 3.3 million transgenic male mosquitoes, and they claim they reduced the wild population by about 80% in the test area 11 weeks later. So they did a pre-count of the mosquitoes before they released the transgenic males. And then after 11 weeks, they surveyed the population of mosquitoes again in this restricted area on the Cayman Islands in 2009. So what are some of the good and bad aspects of this riddle technology? Well, the good aspect is it's a self-limiting system. So the idea is you want the genetically modified organism to die out. And since these mosquitoes are carrying a lethal gene, they're not going to persist in the environment. But this idea that it's a self-limiting system is also not so great, in that you have to continually raise millions of these mosquitoes, and you have to continually release them instead of only having to do it once. And this, of course, makes it much more expensive. So this is the field site in the Cayman Islands where Oxitec conducted this experiment. It was fairly small, 16 hectares, 40 acres. And again, they assessed the mosquito populations in this small area before they released their transgenic mosquitoes, and then 11 weeks later. And of course, choosing an isolated island is a convenient experimental site for releasing these transgenic mosquitoes to limit their potential spread. So again, just a review of how this riddle technology works in general. You have a riddle male that's carrying that dominant lethal gene. Or in that population, you'll have wild-type males that aren't carrying that gene. But the riddle males that are mating with the wild-type females, they're going to produce viable eggs. In this case, it's different than when radiation was used to sterilize the insects. The females are going to release viable eggs. Those eggs are going to hatch into larvae. However, the late-stage larvae and the pupae do not survive. And this is when that lethal gene kicks in, at the late stage, larval stage, and the pupal stage. Here you'll see also no tetracycline. So this antibody tetracycline, it's not available in the environment, but in the laboratory it can be supplied during rearing, and this prevents the action of that dominant lethal gene so that you can actually rear these transgenic mosquitoes in the laboratory in the presence of tetracycline, and then you can release them in the environment. They can do their job, but then in the absence of tetracycline, when that dominant lethal gene will die out with the mosquitoes. So I mentioned that Oxitec, obviously this has a lot of potential both as a business and to solve some important insect diseases. They've conducted trials in other areas of the world. So the Cayman Islands, that was 2019. In 2015, they conducted a trial in Brazil. And of course, for marketing, they're now calling these mosquitoes friendly. Aedes aegypti. And in this trial in Brazil, they're claiming that in this case, they didn't look just at the reduction of the mosquito population. They looked at whether or not they were able to reduce the occurrence of the disease. And so in this trial, they're claiming that they reduced the mosquito population by 95% in a particular neighborhood. And then they're also claiming that compared to the year prior, the year during which they reduced the mosquito population in this neighborhood, there was a 91% decrease in dengue fever. So again, they're trying to build a body of research to support the continued development of these genetically modified mosquitoes, with the hope of being able to eventually use this in a wide scale globally. Of course, the next step is to conduct trials in the United States. So Oxitec has been looking at Florida and Zika as a disease. And at this point, Oxitec has continued to seek approval from the EPA and other regulatory bodies to conduct this trial in Florida. And I think it was just this spring that Oxitec has received permission to do a trial in 2021, so next year, in the Florida Keys. All right, so Oxitec has developed this promising technology that's based on the idea of the sterile insect technique. And they've developed this with transgenic insects. We're going to get back to malaria. And I mentioned earlier in the lecture that the rate of technology changing is amazing right now. And so again, we've had this development of CRISPR-Cas. I'm sure you've heard about this in your lectures. CRISPR-Cas has made it very easy to genetically modify a wide range of organisms, including insects and including mosquitoes. So I'm going to switch now to another technology to finish this lecture. And this is something that I think everyone, whether or not you're interested in biotechnology or entomology, I think it's very important to be aware of this technology, since it's going to have a significant impact on our future. And so we're going to look at this idea of a gene drive and how CRISPR-Cas has been used to create a gene drive in order to manage mosquitoes. So this gene drive idea is, well, the concept has been around for a while, but the ability to actually demonstrate it in a laboratory has been more recent. And again, you know, it's pretty amazing technology based on the abilities that we're now able to do with CRISPR-Cas. So again, I'm going to show you a couple of videos because I think they really capture this very nicely.
A gene drive guarantees that a specific gene will be inherited. Gene drives are attached to the chosen gene and put into the organism. One gene can have different versions. Each organism gets 2 copies of each gene. Without a gene drive, when parents with different versions of a gene pass on their DNA, each version gets inherited by only half of the offspring. With a gene drive, when parents with different versions of a gene pass on their DNA, essentially all offspring will inherit the gene with the drive, generation after generation after generation. A gene drive contains instructions for a molecular tool that targets other versions of the chosen gene. The tool scans the organism's DNA and looks for other versions of the gene. Once it finds another version, the tool cuts it out. Left with a hole, the body uses the gene with the gene drive as a template to patch it up. The organism now has 2 copies to pass on to the next generation.
We're going to finish up the lecture with a TED Talk video, and it's about a researcher that had been working many years to solve this problem of mosquitoes vectoring malaria, and it kind of illustrates that the point of technology change. And this talk will describe how this researcher had been working for many years on some of these other biotechnology approaches, and then along came this new technology, CRISPR-Cas, and this idea of a gene drive, and how that dramatically changed the research and what they were able to accomplish in the laboratory. So we'll go ahead and take a look at that now.
Gene drives. I'm going to start by telling you a brief story. 20 years ago, a biologist named Anthony James got obsessed with the idea of making mosquitoes that didn't transmit malaria. It was a great idea and pretty much a complete failure. For one thing, it turned out to be really hard to make a malaria-resistant mosquito. James managed it finally, just a few years ago, by adding some genes that make it impossible for the malaria parasite to survive inside the mosquito. But that just created another problem. Now that you've got a malaria-resistant mosquito, how do you get it to replace all the malaria-carrying mosquitoes? There were a couple options, but plan A was basically to breed up a bunch of the new genetically engineered mosquitoes, release them into the wild, and hope that they pass on their genes. The problem was that you'd have to release literally 10 times the number of native mosquitoes for that to work. So in a village with 10,000 mosquitoes, you'd release an extra 100,000. As you might guess, this was not a very popular strategy with the villagers. Then, last January, Anthony James got an email from a biologist named Ethan Beer. Beer said that he and his grad student, Valentino Gantz, had stumbled on a tool that could not only guarantee that a particular genetic trait would be inherited, but that would spread incredibly quickly. If they were right, it would basically solve the problem that James had been working on for 20 years. As a test, they engineered 2 mosquitoes to carry the anti-malaria gene and also this new tool, a gene drive, which I'll explain in a minute. Finally, they set it up so that any mosquitoes that inherited the anti-malaria gene wouldn't have the usual white eyes, but would instead have red eyes. That was pretty much just for convenience, so they could tell at a glance which was which. So they took their 2 anti-malarial red-eyed mosquitoes, put them in a box with 30 ordinary white-eyed ones, and let them breed. In 2 generations, those had produced 3,800 grandchildren. That is not the surprising part. This is the surprising part. Given that you started with just 2 red-eyed mosquitoes and 30 white-eyed ones, you'd expect mostly white-eyed descendants. Instead, when James opened the box, all 3,800 mosquitoes had red eyes. When I asked Ethan Beer about this moment, he became so excited that he was literally shouting into the phone. That's because getting only red-eyed mosquitoes violates a rule that is the absolute cornerstone of biology, Mendelian genetics. I'll keep this quick, but Mendelian genetics says that when a male and female mate, their baby inherits half of its DNA from each parent. So if our original mosquito was AA and our new mosquito was AB, where B is the anti-malaria gene, the baby should come out in 4 permutations: AA, AB, AA, BA. Instead, with a new gene drive, they all came out AB. Biologically, that shouldn't even be possible. So what happened? The first thing that happened was the arrival of a gene editing tool known as CRISPR in 2012. Many of you have probably heard about CRISPR, so I'll just say briefly that CRISPR is a tool that allows researchers to edit genes very precisely and easily and quickly. It does this by harnessing a mechanism that already existed in bacteria. Basically, there's a protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. The result is basically a word processor for genes. You can take an entire gene out, put one in, or even edit just a single letter within a gene. And you can do it in nearly any species. Okay, remember how I said that gene drives originally had 2 problems? The first was that it was hard to engineer a mosquito to be malaria resistant. That's basically gone now, thanks to CRISPR. But the other problem was logistical. How do you get your trait to spread? This is where it gets clever. A couple years ago, a biologist at Harvard named Kevin Esvelt wondered what would happen if you made it so that CRISPR inserted not only your new gene, but also the machinery that does the cutting and pasting. In other words, what if CRISPR also copied and pasted itself? You'd end up with a perpetual motion machine for gene editing. And that's exactly what happened. This CRISPR gene drive that Esvelt created not only guarantees that a trait will get passed on, but if it's used in the germline cells, it will automatically copy and paste your new gene into both chromosomes of every single individual. It's like a global search and replace, or in science terms, it makes a heterozygous trait homozygous. So, what does this mean? For one thing, it means that we have a very powerful, but also somewhat alarming new tool. Up until now, the fact that gene drives didn't work very well was actually kind of a relief. Normally, when we mess around with an organism's genes, we make that thing less evolutionarily fit. So biologists can make all the mutant fruit flies they want without worrying about it. If some escape, natural selection just takes care of them. What's remarkable and powerful and frightening about gene drives is that that will no longer be true. Assuming that your trait does not have a big evolutionary handicap, like a mosquito that can't fly, the CRISPR-based gene drive will spread the change relentlessly until it is in every single individual in the population. Now, it isn't easy to make a gene drive that works that well, but James and Esveld think that we can. The good news is that this opens the door to some remarkable things. If you put an anti-malarial gene drive in just 1% of Anopheles mosquitoes, the species that transmits malaria, researchers estimate that it would spread to the entire population in a year. So in a year, you could virtually eliminate malaria. In practice, we're still a few years out from being able to do that, but still, 1,000 children a day die of malaria. In a year, that number could be almost zero. The same goes for dengue fever, chikungunya, yellow fever. And it gets better. Say you want to get rid of an invasive species, like get Asian carp out of the Great Lakes. All you have to do is release a gene drive that makes the fish produce only male offspring. In a few generations, there'll be no females left. No more carp. In theory, this means we could restore hundreds of native species that have been pushed to the brink. Okay, that's the good news. This is the bad news. Gene drives are so effective that even an accidental release could change an entire species, and often very quickly. Anthony James took good precautions. He bred his mosquitoes in a biocontainment lab, and he also used a species that's not native to the US, so that even if some did escape, they'd just die off. There'd be nothing for them to mate with. But it's also true that if a dozen Asian carp with the all-male gene drive accidentally got carried from the Great Lakes back to Asia, they could potentially wipe out the native Asian carp population. And that's not so unlikely, given how connected our world is. In fact, it's why we have an invasive species problem. And that's fish. Things like mosquitoes and fruit flies, there's literally no way to contain them. They cross borders and oceans all the time. Okay, the other piece of bad news is that a gene drive might not stay confined to what we call the target species. That's because of gene flow, which is basically a fancy way of saying that neighboring species sometimes interbreed. If that happens, it's possible a gene drive could cross over, like Asian carp could infect some other kind of carp. That's not so bad if your drive just promotes a trait, like eye color. In fact, there's a decent chance that we'll see a wave of very weird fruit flies in the near future. But it could be a disaster if your drive is designed to eliminate the species entirely. The last worrisome thing is that the technology to do this, to genetically engineer an organism and include a gene drive, is something that basically any lab in the world can do. An undergraduate can do it. A talented high schooler with some equipment can do it. Now, I'm guessing that this sounds terrifying. Interestingly, though, nearly every scientist I talked to seemed to think that gene drives were not actually that frightening or dangerous. Partly that's because they believe that scientists will be very cautious and responsible about using them. So far, that's been true. But gene drives also have some actual limitations. So for one thing, they work only in sexually reproducing species, so thank goodness, they can't be used to engineer viruses or bacteria. Also, the trait spreads only with each successive generation. So changing or eliminating a population is practical only if that species has a fast reproductive cycle.
All right. Now that you can see the tremendous potential of this technology and also some of the concerns that this technology brings, I'm just going to leave you with this parting thought. Should we extinct mosquitoes? What are your views on transgenic insects? Are you for it? Are you against it? I guess I'd just like to leave you with the thought of this new technology, this idea of a gene drive and how it might be used for good purposes, but how it could also escape and be problematic. And so with that, I'll conclude this lecture. Thank you.